Welcome again. This is part 2, where we're going to talk about the data you submit, and resources to help you. I want to talk about the issues of quality of the streamlined data. We believe the data that is used in this competition is nearly the best quality data collected of this type, but it still contains noise. There are limitations because we are imaging live humans, which means there may be low signal noise at any given location. Humans have a limited tolerance for how long they can be scanned. They move, things like that limit the quality of the data collected. Also, there are implications of this noise to the data mining techniques, that some percentage of the fiber streamlines are errors in the data set that we have. It's not perfect data. Errors may include false fibers, false turns, missing fibers, false terminations in the data set. In the expert coding, the expert has tried to remove those from the test set that we're dealing with. In the competition, we require you to map the 20 to 50 tracks, and we'll score 80% of those fibers and hope that there are a few noise fibers in that set. This illustrates some example of the noise fibers. So we often have these sort of single far-ranging fibers, as illustrated by the yellow arrows here, that are typically errors. A second problem that we have is false continuation. That is, one fiber track went into an area and another fiber track came out of the same area, and the fiber tracking might connect those as a single fiber. Another element is ending in not gray matter or subcortical tissue. If a fiber is sort of ending in midair, you know it's wrong. I note that in fiber tracking of other data sets, you'll find other problems such as incoherent direction of the fibers or running along the cortical surface. We do not find that in this particular, the methods that we have used within that process, but these problems have plagued most previous fiber tracking methods. So in terms of part 2, what is the data that you will receive in terms of the data and formats? What do you submit for each of the challenges? A little bit of information about the getting starting of the data sets, and some approaches to the problems and the solutions. I note that there are many metrics that you can explore within that set. Each of the fibers, you have a length, center of mass, a geometric center of mass, fiber trajectory, surface contact, similarity to nearby fibers within that process. For each of the challenges, you'll need to identify a reference fiber track, some fiber going through the area that will define that track. We'll then compare that with metrics of the bundle, such as the mean track fiber length, its clustering, the similarity of the fibers to that target track that you're dealing with. There are a variety of metrics that we could use. You could be basing things on a single reference fiber, on a mean fiber, as illustrated by the yellow fiber on the right, perhaps a mean trunk fiber, that is, exclude the initial and final turns of the fiber track and match the middle point within that. All these are possible metrics that you might want to explore in your entry. The data, a fiber is a 3D curve in space, which can be defined as a set of 3D points in terms of the XYZ points. These 3D curves are macroscopic, representing the diffusion direction of the white matter fiber tracks in the brain. Mathematically, a fiber is a set of number of points in this data set at 1 millimeter distance boundaries within that set, then what you have is, for each of those points, the x and y and z point in a normalized brain position for that fiber. We've calculated for each of the brains 300,000 fibers. So for the white matter, for the whole brain, it's a set of these 300,000 fiber tracks that we're working with. A fiber bundle can similarly be defined as a subset of those 300,000 fibers that we're dealing with. So you're going to be going from the axon streamlines, 300,000 fiber tracks on the left, to 20 to 50 fiber tracks to identify the tracks of an individual. Now I want to note some properties of the fiber tracking. Similar fibers start and end in the same cortical area. However, there are exceptions to this rule where the same cortical area projects to two different areas. So this is a useful feature, but by no means necessary and sufficient. Similarly, similar fibers typically have a similar length, so you would cluster on the length within this process. But again, it's not always true. Sometimes you have fiber bundles of the same length going to different areas, sometimes even with the same center of mass. Similarly, we may have a geometric center of mass for a set of fibers, which is often a good discriminant about a fiber bundle, but this too is not always true. One of the metrics that is useful is the distance between two fibers along the length, and this is an illustration from O'Donnell of how one might calculate those distances. There are many possible fiber similarity metrics. The fiber streamlines are a set of points, and it can be characterized based on metrics such as the length, center of mass, geometric center of mass, volume, laterality, and curvature. The ones colored in green are ones that we've pre-computed for you. Metrics along the fiber are the position, the curvature, the tangent, the fractional, and isotropy. That is how much the fibers are representative of a single bundle, and the apparent diffusion constant for the flow of water within the fiber. Then you have the segmentation within the fiber track, things such as the maximum length, without a sharp turns, the midsection, the 20 to 50%, things like that. In terms of similarity metrics, there are both fiber match similarities, those are the metrics between any two fibers on a single or multiple metrics, and fiber bundle metrics such as coherence within a bundle, bundle size, and shape. Also surface contact issues such as what brain areas does a particular fiber track contact. We will be providing you 6 sets of 300,000 fiber streamlines, providing the fibers in the normalized brain position. And for one brain, we'll give you the expert coding of 8 tracks of that brain. The data set you will send in is basically the assignment of 6 sets of fiber data to streamlines, where 0 means you're not assigning it, and 1 to 50 would be the streamline or the track you are assigning it in. We want you to, the first test is matching an expert, so that you have a data mining segmentation, and that will be compared to our expert segmentation of that data set. So you can think of this as you're going to classify the 300,000 streamlines to match the expert coding of 8 tracks. So you're going to generate the 8 tracks for the data, and then we'll compare it. What you'll actually be submitting in is basically a table which just indicates for each sequential fiber, are you coding it? 0 means no, or if you are coding it, the number 1 to 8 of whether or not it matches is expected to match our expert coding of that fiber track. This concludes part 2 of this tutorial. YouTube requires that we break this into 10-minute segments, so we'll be taking a short break, and there's about a 7-minute section left over. Thank you.